Ladies and gentlemen, good afternoon. My name is Sophie Desplagego and I'm a medical immunologist. My hospital lab is in Marseille, in south of France, and its activity is focused on the exploration of autoimmune and chronic inflammatory disease. First, I would like to thank the organizer for giving me the opportunity to share with you some slides today. We have no disclosure. Infliximab is a chimeric IgG1 monoclonal antibodies that targets TNF. It is highly effective and widely used for treating inflammatory chronic disease like rheumatoid arthritis, psoriatic arthritis, or Crohn's disease. But not all patients have a good response to this treatment. In case of secondary loss of response, the main reason could be the immunization against the drug and the production by the patient of antibodies against the drug. Depending on the studies, the patient could produce these antibodies up to in the 50 cases. This production of antibodies may be related to two kinds of factors: the patient-related factors, as you can see here, such as genetic background, disease stage, or co-treatment, but also drug-related factors like frequency, route of administration, dose of the drug, and also impurities of formulation. Nowadays, ADA levels, anti-drug antibody levels, are included in algorithm for therapeutic drug monitoring of infliximab-treated patients in case of response or in case of non-response. And it has been postulated that formation of ADA increased drug clearance, reduced binding to target, and also increased adverse reaction like allergic reaction during the infliximab infusions. The objective of our study was to evaluate the incidence of infliximab ADAs in a retrospective cohort of both adults, children, and IBD rheumatoid disease patients. So levels of infliximab were also quantified in this study. For this purpose, we used a commercially available ELISA developed by the French company Theradiag. And by using this reagent, we were able to quantify ADA from 10 to 200 nanograms per ml, and infliximab from 0.3 to 16 micrograms per ml. Then we included in a retrospective cohort 872 samples, routinely test and consecutively test in our lab for ADA detection and infliximab through level evaluation. This cohort corresponded to a number of 545 patients with a sex ratio of 2:3, a mean age of 42 years old, with the youngest patient was 5 years old and the oldest was 91 years old. In this cohort, we included 510 patients, adults, and 35 children. On this slide, you can see the results of our study, and you can observe in red that 82 patients out of 545 patients developed ADAs. These rates correspond to 15% of all patients. You can observe that the sex ratio and the mean age of the ADA-positive patient cohorts are similar to those of the whole cohort, and that the positive patient in adults and children cohorts were very similar. Among the ADA-positive patients, we observed that 56 patients suffered from digestive disease, while 35 patients suffered from rheumatic disease. Finally, we found that most of patients who were positive for ADA detection were treated by the biosimilar of Infliximab called Inflectra. In the next step, we looked at the relationship between ADA occurrence in the whole cohort and age or sex of patients, and we found no difference between the occurrence of ADA in function of age or sex of patient. We found on the next slide that most of ADA-positive patients had ADA levels more than 200 nanograms per ml. We can see here there were 51% and didn't receive methotrexate, since you can see here 73 patients out of 82 were not treated by methotrexate. When they receive methotrexate, but it's very small population, we found that they didn't produce less ADA than when they not received methotrexate. Now, here on this graph, we look at the difference in ADA levels in function of the type of chronic inflammatory disease. Either difference between patients suffering from digestive disease or rheumatic disease. And you can see here that the mean values are not statistically different. And we don't find a difference in ADA levels in function of the type of chronic inflammatory disease. Then you look here at the Infliximab serum levels, and on this part of the slide, you can see that for 93 patient samples, out of 104 samples, the levels of Infliximab were undetectable since they were less than 0.3 micrograms per ml. On this graph, you can see that there was no correlation between ADA serum levels and number of Infliximab infusion. Finally, we tried to define some kinetics properties of ADA production. Since 15 patients in our cohort got more than 1 blood samples, and we observed that ADA levels remain stable and are more than 200 nanograms per ml for 9 patients and less than 200 nanograms per ml for 2 patients. On the other hand, ADA levels varied for 4 patients and decreased for 3 patients and increased up to more than 200 nanograms per ml for 1 patient only. In conclusion, we presented here a study including both adults and children and both digestive and rheumatic disease patients, a big cohort, and we found the prevalence of anti-drug antibody about 15%, mostly with level of antibodies more than 200 nanograms per ml. We found that ADA occurrence was not influenced by age or sex of patients, and that ADA levels were not influenced by the type of the chronic inflammatory disease, digestive or rheumatic, the number of Infliximab infusions, or not influenced by taking methotrexate. Moreover, the level of infliximab were less than 0.3 microgram per ml for 93 patients out of 104 ADA-positive samples. ADA levels remain stable for 9 out of 15 patients with immunological follow-up. Thank you for your attention.